different kinds of cells there are in a human body? 1, 10, 1 million? In fact, a human being is made out of approximately 200 different types of cells. These allow us to walk, eat, breathe, think, and perform all our daily activities. But where do these cells come from? It all starts here in the uterus with only 2 cells, the sperm and the ovum. When the ovum is fertilized, it starts dividing into identical unspecialized cells. After the first divisions, the group of cells is called morula. These cells are totipotent cells, meaning they do not carry out any direct function, but have the potential to become any of the cells of the human body or placenta. The morula cells keep dividing, and after 4 days they form the blastocyst. This is made up of 2 different kinds of cells. The outermost layer is going to form the placenta, while the inner part is going to give rise to all the cells in the human body. These are embryonic stem cells, and they have 2 main characteristics. Theoretically, they can divide an infinite amount of times, giving rise to identical pluripotent stem cells. Alternatively, they can divide to form 2 more specialized cells, which go on to divide again and again to form even more specialized cells. Let's now look closely at the differentiation process of an embryonic stem cell to form a neural cell. This is a pluripotent embryonic stem cell. It can differentiate into any cell. If, however, it divides into a neural stem cell, it will become multipotent, meaning it will not be able to give rise to anything other than neural cells. The multipotent cell then has the choice to divide either into a glial precursor cell or into a neuronal precursor cell. Whichever path it chooses, it will lose some of its potency because a glial precursor will not be able to generate a neuron, and the neuronal precursor will not be able to divide into a glial cell. Then, the glial precursor will further differentiate into an astrocyte or oligodendrocyte, which are both glial cells, while the neuronal precursor will become one of the different types of neurons. These are the 3 main components of the central nervous system. Each of them carry an important function in the central nervous system, and the lack of any of them causes serious damages to the functioning of the human organism. The neuron is the core component of the nervous system. Its function is to receive signals in the form of impulses and to transmit them, usually to other neurons, through synapses. A neuron receives the impulse through many branch projections connected to one end of the cell body, called dendrites. Then, the signal goes through the neuronal body, where the nucleus is located, and the impulse is transmitted to another cell through the axon, a single long projection. The astrocyte is an auxiliary cell of the neurons in the central nervous system. Astrocytes provide the neurons with nutrients, maintain the extracellular ion balance, ingest the injured neurons, and perform other tasks for the neural tissue's health. The oligodendrocyte is a glial cell present only in the central nervous system. It secretes a layer of myelin around the axons of the neurons, which speeds up the impulses and makes the synapses more efficient. The process of differentiation of an embryonic stem cell into neural cells still has many unknown factors, and scientists are trying to find protocols to reliably differentiate the stem cells into the desired unipotent cells. The embryonic stem cells the scientists work with are extracted from the inner part of the blastocyst. These are usually placed in a medium and are allowed to grow undifferentiated on a feeder cell layer. Then they are transferred into different media depending upon which kind of cells scientists want to obtain, and they grow and differentiate in it in 2 dimensions floating in the medium. The aim of this project was to differentiate mouse embryonic stem cells into 3-dimensional neural tissues using a protocol tested for the differentiation of stem cells in medium. Embryonic stem cells were used from mouse uteri instead of human. This made the growing and differentiating process faster. The embryonic stem cells were taken from 2 different types of mice called D3 and CGR8. D3 cells are common mouse embryonic stem cells which need a feeder cell layer to grow on. CGR8 cells are also embryonic stem cells, but they do not need the feeder cells to grow. These 2 cell lines were separately grown and then placed in the form of droplets on a membrane. The medium to differentiate the stem cells into neural cells was added beneath the membrane. This allowed the cells to be in contact with the air from above and with the medium from below. The cells could therefore grow in close contact with each other, and the medium could be changed without touching the cells. After 3 weeks of growth and differentiation, the droplets of cells became tissues. While they were growing, photos of their external appearance were taken. After 2 days, the cells in both D3 and CGR8 tissues were evenly distributed, but already 2 different patterns could be distinguished. After 8 days, the patterns of the 2 tissues changed. On the D3 tissue, surface circles could be noticed, while a dark brown substance was present on the CGR8 tissues. After the first week, the pattern remained constant. After 3 weeks of growth, the tissues were embedded in paraffin. Then, the paraffin with the tissues inside was cut perpendicular to the membranes in thin sections which were placed on slides. After this step, the sections of the tissues were ready for analysis to determine what kinds of cells were present in them. To determine the morphology, some of the sections were stained with crescel violet staining, which marked the nuclei of all the cells. Under microscopic analysis, a tissue section from the D3 cell line looked like this. The grey layer on the bottom is the membrane where the cells grew on. The purple dots are the nuclei of the cells, while the transparent parts between them are all the other organelles of the cells and the cytoplasm. The cells in this section are evenly distributed and in close contact with one another. It is possible to notice a jelly-like layer at the air border. This is formed by the axons of the neurons which grouped together. The section of the CGR8 tissue, stained in crescel violet, looked completely different. First of all, the distribution of the nuclei is not homogeneous, and there are regions where there is a very high concentration of cytoplasm, which is not typical for neural cells. Some circular agglomerate of cells can also be noticed, which are a sign of gland cells. These are close to round nuclei-free regions, which represent the substance secreted by the gland in the tissue. Additionally, CGR8 sections also contain regions of dead cells, shown by the smaller fragments of nuclei. CGR8 tissues also showed signs of cartilage. This first crescel violet analysis showed the morphological differences between the CGR8 and D3 tissue and gave the first indication that the CGR8 cells were not only neural. Subsequently, different immunohistochemical analyses were performed. Immunohistochemistry is a technique used to detect a certain protein in a tissue. Each different type of cells produces some proteins which are not produced by any other type of cells. If one kind of these specific proteins is injected into an animal, for example a goat, the goat's organism will produce antibodies to detect and fight the unknown protein. These antibodies can be collected and poured on a tissue sample. If the tissue contains a specific protein, the goat antibody will bind to it. Then, a second fluorescent antibody, which detects the goat's antibodies, can be poured on the tissue. The secondary antibodies will bind to the goat's antibodies. Then, under the fluorescent microscope, the secondary antibodies will emit a color and mark the place where the primary antibodies and the specific proteins are. As these proteins were specific to only one type of cell, by knowing the presence, the amount, and the position in the tissue of the protein, the presence, the amount, and the position of the cells are also known. In this project, each section was stained with 3 different fluorescent antibodies, 2 of which marked different cells each time, and 1 of which always marked the nuclei of all the cells. First, the presence of neurons and neuronal precursors were analysed by immunohistochemical analysis. It can be seen that the majority of the cells in the D3 section are neurons. The section is almost completely stained in red, and there are very few purely blue dots, which represent the nuclei of the cells that are not neurons. The neuronal precursors are very few. This shows that the differentiation process was almost completed. In the CGR8 tissue, the neurons are not evenly spread and are inferior with respect to the other types of cells. There are no neuronal precursors. Thus, the differentiation process in the CGR8 cells ended faster. There is an empty black space in the center of the tissue. This is the secretion of a gland observed by the crescel violet staining, and therefore this region does not have any nuclei. Subsequently, markers were used to verify the presence of neurons and astrocytes in the tissues. In the D3 tissue, astrocytes are present even if the neurons are the most common cells. The astrocytes are spread quite evenly around the tissue. If the slide is further magnified, it can be noticed that near the air surface, the astrocytes overlap and follow the path of the neurons. This shows that the astrocytes in this tissue interact with the neurons as they do in vivo. Near the border with the air, there is a layer where there are no blue nuclei, but where the red colour is continuous and very intense. These are the axons of the nuclei, previously noticed by the crescel violet staining. In the CGR8 section, the neurons are mainly located near the air surface. There are no visible long agglomerates of their axons, and there is no evidence that they interact with the astrocytes. The latter are spread evenly around the section with the exception of the clearly marked gland cell and its secretion. The astrocytes are the predominant cells in the CGR8 tissue. The tissues were also tested for the presence of oligodendrocytes and mature neurons. The D3 tissue had substantial amounts of oligodendrocytes, which produced a layer of myelin around the mature neurons, thus reproducing their functioning in vivo. No cell of this kind was found in the CGR8 sections. The tissues were tested for the presence of synapses. It was found that in the D3 tissue, there were synapses near the surface with the air where previously the axons agglomerate was found. In contrast, there were no synapses in the CGR8 tissue. To find a reason for the lack of neuronal activity, the CGR8 tissue was tested for the presence of mature neurons. The analysis found that there were very few, almost 0 mature neurons, showed by the red colour. This indicates that the neurons died in the process of cell growth. The dead neurons were detected by the crescel violet staining. In this project, 2 mouse embryonic stem cell lines were comparatively grown and the tissues they formed analysed. The aim of the investigation was to find which cell line more efficiently produces neural tissues and what neural cells they differentiate in. It was found that the D3 cell line differentiates only into neural cells. The majority of the cells of this tissue were neurons. However, astrocytes and oligodendrocytes were also found. The cells appeared to be healthy, and the cells in vitro behaved analogously of the cells in vivo. Astrocytes interacted with neurons, oligodendrocytes made the myelin sheath for the axons of the mature neurons, and the neurons transmitted impulses to each other and grouped together their axons to form nerves. The tissue obtained from CGR8 cells contained more than just neural cells, and therefore cannot be called neural. Neurons were present even if they were not as abundant as the astrocytes, but most of them died before reaching maturity. No oligodendrocyte was detected in the tissue. Altogether, the accomplished study provided a significant amount of evidence that the D3 mouse embryonic stem cells are more suitable and reliable than CGR8 cells for the production of healthy and active neural cells. Therefore, D3 rather than CGR8 cells have significant potential for further biotechnological and biomedical applications as drug testing.